here at Flexergy, we're always excited to keep learning and also keep understanding what's going on with the product and the impact we're making, but also any issues that come up. So today, our goal is going to be to introduce the app science team, kind of our problem solvers here at Flexergy, and then Dr. P may have a little bit of content, but also it's an open forum to ask some questions. So we're going to have this session every 2 months, and then we should have a webinar on a specific topic in between. But yeah, this is, I think, we may have a smaller group today, but I think that's good. We can get through some of the, specific topics. And again, this is also to see maybe what topics we need to present on moving forward. So I will hand the mic over maybe to Andre, one of our application scientists here at Flexergy. He'll give a quick introduction and talk about one of the problems they solved, and then we'll let it go from there.
Thank you, Eric. I'm going to share my screen and we're going to go through a short presentation. All right. So who is the application science team? It's myself, Andre, and my colleague Selena, who is also here in the room with me. We both have molecular biology and, in Selena's case, molecular microbiology research experience. Selena studied sea turtles in Honduras, and I happen to like plants a lot. But what is an application scientist? So basically, we work to apply scientific principles and techniques to real-world problems, and we are the liaisons between Flexergy users and Flexergy. We support customer support, research and development, regulatory, sales, and marketing internally. And we make sure that the voice of the customer gets to everyone involved in the company to some degree. So yeah. That's part of what we do. And this goes more into detail about what we do. We have our market requirements. We conduct research, products and processes, technical support, training, both internal and external, and account management. So we're here to provide solutions to your issues, your problems, and to create new solutions for whatever you see as a hurdle in your day-to-day. So what does that translate to? Lots of projects. So these are some of them. This is things we do day to day. We wear many hats, but at the end of the day, we're here to support the customer and to make sure the customer is heard within Flexergy and that Flexergy can respond as a group to the needs of the customer. So we successfully converted a clinic from using a 6-day culturing protocol to a 3-day protocol using a combination of culturing and Flexergy. Their issue was long turnaround times and high costs associated with the culturing method. And what we did was a comparison study between culture and Flexergy PCR. We helped support that. Selena, she was very involved in that. And as a result, the site fully switched to Flexergy with some slight modifications. This is what they were doing previously. I think you can all see the screen over here. They did 1, 2, 3, 4, 5 overnight incubations. So this allows for very good specificity, as well as good sensitivity. However, it takes a minimum of 6 days to get those results. Now, that has a lot of technician time, a lot of resources, and difficult to perform. So what did they do in the meantime when they evaluated us? Well, they took a Flexergy PCR test after each incubation, and they also did an XLT4 plate selection step after the selenite broth incubation, that's a Fluxergy step, just to confirm that what we're catching is indeed Salmonella. After this was concluded, they decided to add an additional step in front of the solenoid enrichment broth, which is buffered peptone water. And that adds an extra day to the Fluxergy process. However, if you look at this chart, you'll still see that on the third day, all that it takes is running the Fluxergy Salmonella PCR, which is 5 minutes of prep time and an hour or so hands-off time while the PCR runs. So all in all, it has been a very successful transition from their previous culturing method for environmental samples into the Fluxergy method of doing the same thing. Just a lot less time and a lot less man hours. All right, I'm going to stop sharing.
That's great, Andre. It actually segues into a question I think I hadn't thought of until I got involved in these conversations, particularly with Dr. P, is the need and the value of, if you're doing culture and PCR simultaneously, making sure that you're comparing the same thing. So, Dr. P, we've talked about this, making sure that it's the same sample that's being tested. It's the enriched sample that's being tested, not the original sample, because of the distinctions. And I think that's a really important when we're running them in parallel to sort of expand on.
Absolutely. We need to be comparing the same thing at each step of the way.
Hey, Andre, may I ask how the results compared if you look at conventional culture with the various steps? I mean, this is a very comprehensive culture panel that they did in that clinic. Kudos to them. But as you can see, it definitely extends the testing timeline. Did you look at the outcome results? In other words, how much agreement did you have between the conventional culture versus Fluxergy? I don't know if you have the data set.
Yeah, I do have the data. 1 second, let me just.
You don't have to show it. You don't have to show it. Just by memory, if you know what was the overall agreement and where you may have discrepancy where you feel that PCR may have maybe detected more positive compared to conventional culture.
Of course. So I have the data in front of me, and after they moved to the new protocol, they've run 24 tests with 69 tests, apologies, with 100% agreement with the culture method.
So same detection rate, just faster, basically.
Correct. And so the big extensive protocol, which I showed initially, with that one, it was 69% agreement, which is why we decided to change to Fluxergy and to give that new protocol a chance, because the prior agreement was not that great, and we decided to see why. And once we determined that equivalency in the transition phase, the agreement afterwards was at 100% for those 69 samples.
Based on the results, would you recommend that pre-enrichment step with buffered peptone water on environmental sample? Or would you skip it?
Personally, I would skip it. However, they included it for additional peace of mind. I don't feel that it's necessary. However, everybody is welcome to adopt the protocol to whatever they feel their needs are, and to adapt to whatever their process is. So I don't think it's necessary. However, it does give peace of mind to the clinic.
It'll be interesting to compare, the step just going into selenite broth versus that pre-enrichment step. I mean, it's kind of interesting, you know, all these steps are based on culture. The majority, that's something that was interesting when we gave that webinar with Dr. Sykes. You start to realize that all the data that we have, the epi data, the, you know, data on, you know, how to recognize how many sample can you collect, what's, you know, the relationship with pooling, it all relates to culture, conventional culture. And I always wonder, how would that data look if we would repeat it and use PCR? And I bet you would look different to the advantage of PCR, meaning that would probably be faster and have more positive result, which, you know, it's not necessarily what people want. But, you know, the buffer peptone water step is generally used as a way to wash the sample. I mean, this is a large volume they use with a wipe. So you can't just put like a, I don't know if you've ever seen them. I mean, these things are huge. You can't just put them in a, you know, 20 ml tube. That wouldn't work. So I think that the idea of getting a large surface sample to mix it with something, and it doesn't really matter if it's, you know, PBS, saline, or buffer peptone water, and then once you have mixed it and you shake it or vortex it or throw it on the ground for a few times or step on it, whatever you decide is more, you know, relevant and fun to do. But after that is to take an aliquot and then inoculate your selenite broth. I think that's that step for, like, again, a large surface collection device, I would say it's a must. Now, it's different when we collect the environmental sample with the sponge. The sponge is a very small surface. I mean, it's, you know, I would just guess it's probably, you know, 1.5 over 3 inches, and it has both sides, but it's, you know, it's probably not as surface friendly as a Swiffer wipe, and it's definitely not as surface friendly as the drag swabs. This is what we use in the hospital. We use, you know, they look like Q-tips on a wooden stick, and you take 2 or 3 in your hand and you just get in the corner or the drain and then put them in the bag. So I think it's, you know, it's just different protocols out there. You got to use the protocol that you feel comfortable, that gives the best and most reliable result for your clinic. But that's, I'm very excited to see that data set. So good work there, Andre.
So there's potentially value in having the wash there, but not necessarily the need for that first incubation step, is what you're saying. So you have the wash there to basically elucidate the salmonella out of the sampling matrix into the liquid, and then you're incubating directly from there.
Yeah, exactly. So the benefit is to take that large surface sample. I mean, it's a, you know, it's a piece of sheet. That's what it is. So imagine if you now fold it and you stick it in that little selenite tube, it's gonna be, I don't think you can do it. But if you take that sheet and you put it in a, you know, 1 gallon Ziploc bag and add 20, 30, or 40 milliliters or whatever it is, it just happen to be, you know, buffer peptone water, use saline, and then you really shake it. That really dissolves all the organic material and all the material that is attached on that wipe and puts it in a solution. And then you take an aliquot. That would be my strategy. And it's actually a strategy that, you know, there was one question here about, and I'm sorry, I'm jumping around if I'm not, we have no script. So, and they cannot stop us. So that's the good thing. But one protocol that we looked at, and it's a question that I see here from one of the participants, is, you know, the rate of negatives, or excuse me, the rate of errors when you're testing feces. And I think it goes a lot with, you know, the dilution effect. And the way I personally would do it is get maybe a larger volume of feces, mix it with a solute, and that can be saline. I mean, if you have nothing else than water, then use, you know, water, as long as the water isn't contaminated with salmonella. You can use buffer peptone water. And the idea is to then shake it. And it has 2 advantages. The first of all, you can take a larger volume to start with. We know that, you know, 10 grams of feces will yield better result than 1 gram of feces. There's no benefit above 10 grams of feces. And that's what I'm saying. It's all relates to culture outcome. That's why, you know, we need to look at that with a grain of salt. But so the first advantage, you start with a larger amount, so greater weight of feces, and then you shake it, and we know that salmonella in the feces is not homogeneously distributed. It has colony clusters. So the idea is, if you have a cluster in that 10 grams, now that cluster is in the solution, and then you take a certain amount, I would say, perhaps 1 milliliter of that 10 gram of feces in PBS, and then inoculate the selenite. That's the protocol that I would endorse, which is slightly different, because where I see the difficulty is, well, what is 1 gram of feces? Show me 1 gram of feces. I'm like, yeah, it's a little bit, and sometimes a little bit is too big. The other benefit of diluting that initial sample, and it doesn't matter if it's an environmental sample or fecal sample, is that you dilute the inhibitors, and that's mostly true for feces. And one of the reasons why we get errors not frequently, but with a higher rate with fecal sample, is that because in the inherent inhibition of feces, which then is not removed through the PCR process. So that, that's something we kind of have to follow and look at, and maybe improve and adapt the protocol. Sorry to give you more headaches, Eric, but that's definitely something that, if that becomes a problem, it's, what is an error rate, acceptable error rate? And I would say 5% and less, I would be happy with that. But if it gets in the, higher than that, and the error is the same, and the error comes from inhibition, we have to look at, what is the error code? So, we need to talk apples. But if the error code is that the internal control isn't recognized because there is inhibition to that enzymatic process of PCR, then we need to dilute the sample. The best way to dilute it is at the beginning.
Yeah, and I'll just jump in quickly. So no, I think the 5% is actually a great number. I think with all of our testing right now, we're just under 5% with all of our clinics. So we kind of see that data. So that's good to see, but it's definitely something that requires some reps. And I don't know if Andre or Selena, are you able to maybe comment a little bit about what you see in the field in terms of why a group may have more errors? I think, yeah, the protocol can always be optimized too, but it seems like we've been able to get the error rate down under 5% with many of them testing fecal samples. So just want to get your opinion there, since you're looking at the data every day.
Of course. The two big things to note, ways to avoid errors, are, first of all, being diligent with your sample processing and with your pipetting. We understand that you might be in a rush. You might want to get the results out as quickly as possible, but taking that extra second to make sure you don't have bubbles or debris in the pipette is huge. If you avoid that, you'll avoid the majority of errors. However, there are instances where you might do everything perfectly and you still get inhibition. However, there is no way to tell that until you actually run the sample. And something to keep in mind is more is not always better. When you're collecting sample, getting a really dirty sample is more prone to errors than getting one that's more free of debris. Those are just some of the things that Selena and I and the team see in terms of errors and some of the common causes of errors.
I think that comment, more is not better, is an interesting one. And again, these are things I hadn't thought of until I've been engaged in these conversations. But that dilutional effect, that capacity to dilute the inhibitors, but relying on the exquisite sensitivity of PCR, it's somewhat counterintuitive to think that you're going to dilute the sample and make it better. But it really is that interplay between diluting the bad, but the test is so exquisitely sensitive that as long as it's there, it will be found. And it's understanding that counterintuitive nature of it, that in fact less is more in this case, or maybe not less is more, but yeah, that dilutional benefit.
You need to give the enzyme room to work. If you overload the enzyme with inhibitors, it just does not work as intended, and you don't get amplification.
And I think to add there too, it seems like with it's separating the two problems with Dr. P's comment, getting the one colony forming unit, which I think that's what we've kind of determined as our sensitivity. When you enrich the sample, you do maybe need more feces, and it may not be in that small fecal ball. Or we have clinics that are actually pooling fecal balls over 3 days to make sure you have kind of a time period that you're looking at. But then when if you do get that one colony forming unit in our selenite or in the selenite, then you have that enrichment step. So that's where it will grow over that 16 to 24 hour period if it is viable. So it's kind of separating those two parts of like the sample collection, and then okay, what are you enriching, right? So
Absolutely correct. Thank you.
So yeah, not exactly. Yeah, I think it definitely requires some reps and reps to get used to the process there. So yeah, not trivial. Maybe should we knock out the other question there, Dr. P? Let's see. I think we have another question here. Let's see, can we go over the sensitivity and specificity and why we are not determining the neuropathogenic versus non-neuropathogenic strains? I'm assuming for EHV1. So maybe we can talk about the neuropathogenic versus non-neuropathogenic, and also, maybe indication for use and detection of EHV1 in terms of like the conserved genes. I'm happy to go, or maybe we can have Andre go into the sensitivity and specificity of what we've done in terms of our internal validation.
Do you want me to cover that?
Yeah, the
The
neuropathic versus
relevance.
non-neuropathic, I probably feel
Yeah,
more comfortable if you cover that one.
So it's a very unfortunate nomenclature, and I wish they would have never introduced that. So I would say in the mid 2000s, there's a consortium of researchers that defined the entire genome of two EHV1 types. One related to abortion, the other related to an EHM case. And the whole idea was, let's see if from a genetic standpoint, the two strain can be differentiated. Can we find markers that define that a horse will actually develop EHM versus an abortion? And they actually targeted that single nucleotide polymorphism on that polymerase gene that then gave rise to the nomenclature neuropathics, defined as a D752. So it has one different amino acid at position 752 of the amino acid sequence. Way more information that you will ever want to know. So I hope you'll be able to sleep tonight. But that single nucleotide changes the amino acid, single amino acid, which they change the conformation of the protein. And that DNA polymerase is a protein, as the name says, is essential for the replication of the virus. So the concept was brilliant. It's like that single nucleotide polymorphism changes the biology of that strain, makes it more virulent. Hence, the outcome is EHM versus non-EHM, which by default they call non-neuropathic. So they could have turned it around and call it abortinogenic and non-abortinogenic, but probably wouldn't have been that sexy. Am I right? Unfortunately, if you look at 2007, so we are almost like 18 years later, with the concept that 85% of EHM outbreaks with historical data were caused by the neuropathogenic. So a very nice correlation there. And then if we look at abortion, we were somewhere around 80 to 90% of abortion caused by a non-neuropathogenic. And that's based on screening tens and tens of well-characterized strains. So these are historical strains. So we're looking strains from the 50s, 60s, 70s, 80s, 90s, okay? We recently look at what we call contemporary strain. So strain that circulates, from 2010 to 2023. And if you follow SARS-CoV-2, so COVID-19 and the strain, you know that these suckers they adapt. And so they are RNA viruses, they adapt fairly quickly. But actually they adapt for the good. Their concept is, I want my host to survive. I don't want to kill my host because that's pointless. If I kill my host, I won't get anywhere. So it's been very similar with EHV1. The more virulent one, the neuropathogen out. The virulent one is dying out, and we see that. The virulent one, the non-neuropathogenic, is actually the one that is more prevalent. And these days, 2023 data shows that 80%, 80, apologize for the accent, 80% of EHM outbreak, equine herpes virus 1 malencephalopathy, are caused by a non-neuropathic genotype. That doesn't make any sense, am I right? From a nomenclature standpoint, trying to confuse people, you have an EHM outbreak, and now you're telling me it's caused by a non-neuropathogenic genotype? Sorry. Now, 2 and 2 doesn't equal 4 here. That doesn't make sense. It's because we're really focused on the nomenclature. And so we have made full circle and decided that the genotype is irrelevant. It's really irrelevant from a clinical standpoint, from a management standpoint, from a preventative standpoint. It's irrelevant. What's relevant, however, is that the horse develop or does not develop EHM. That's relevant. There's also a lot of regulation that goes with it. Most states want to know, what genotype is causing that outbreak. And to make everything more complicated, in 2018 out of France and 2021 out of the USA, there is actually a third genotype. So there's the D752 neuropathic, N752 non-neuropathic, and H752. So sorry, there's no name for that one because, 2 names are taken. So we'll call it, non-neuropathic and non-neuropathic. I'm just kidding. We'll call it the new strain. And most of the laboratory in the US will only test for 2 genotypes. So I would say having a test that screen, and I'll stop talking at some point, believe me, a test that specifically target a very conserved area of EHV1, and this is what Floxagy's test does. It targets the glycoprotein B gene. It's a very conserved gene, is present in all the different genotype. It makes more sense because you're gonna associate the result with the clinical entity than trying to go after the genotype. That's the role of the state lab. So don't agonize over that. If in the majority of the state in the US, if you're dealing with an EHM case and it tests positive, let's say on Floxagy, you have the obligation to report that to your state. So these are reportable disease and actionable diseases. Depends a little bit on the state. It's like state of California, I speak for the state of California, anything that is neurologic and is EHV1 positive, independent of the genotype, needs to be reported. So what does the state want? They want to confirm that case. So they'll take an additional sample and they'll run it through the different tests to determine, yes, it is EHV1 positive, and it is caused by either neuropathic, non-neuropathic, or that new variant. It's probably too much information, but in a nutshell, that's what it is. So irrelevant, in my opinion.
I'm glad you answered that question. But no, that's good to know. And I think I can comment a little bit on the sensitivity specificity aspect, at least of our test. I think we're
97.5% for both for EHV1.
Okay, perfect. I mean, and I think just a comment there, I think we're still compiling quite a bit of data with kind of more field studies right now, so that's exciting. But generally, any tests that come out of Floxagy, they go through quite a bit of feasibility testing and development, and then we'll do what's called a method comparison, where we'll compare against, let's say, a reference laboratory assay using clinical samples, either collected from our partner clinics or kindly sent from our partner reference laboratories like UC Davis. Thank you, Dr. P. And essentially, we'll compare our method with that standard or gold standard method. So that's based off our internal testing. I think that was, do you know how many samples that was, Andre?
90.
Okay, so that was 90 samples. So, and I know we're doing quite a bit more work to increase that number and get some publications out. So we should have some more data shortly.
Andre, can you talk on the testing of blood? I mean, I know the test is, look, the test is validated for nasal secretions, but we would be interesting to know if the test could also be used for blood. Do you have any pilot data, any ideas if that would be possible in the future?
I do have some ideas. We have not gotten deep into that testing yet. However, we have noticed that whole blood is a little bit tricky for the PCR. However, if it's diluted a little bit, we're trying to figure out how much dilution it can take and still run with good sensitivity. We're trying to figure that out, but the results are promising. So we are getting to that point where we'll be able to hopefully have a new sample type.
You can let us know the results on Monday, Andre.
I will.
Let's see. Good. I think a few other questions here that we always hear. Maybe for Dr. P or Dr. Sykes, the value of a panel versus single plex. I think I do hear that quite a bit. Hey, I'm already going to send out the panel. Is there value in running a single plex test? Right then. I don't know if you have any comments on that. I have some thoughts, but
yeah, I can dive in there. Look, I think it's not necessarily one or the other. It's when and what information you're trying to derive from that. So, a clinical presentation, and it's the scenario, right? So, if we take the traditional scenario, which is a horse with diarrhea going into isolation, we want to know what pathogens affecting that horse. And there is a number of pathogens that can affect that horse, and there is value in a multiplex. There's no question around that. I think that there's always going to be value in being able to work through a number of differentials. The thing then, though, is that with the rest of our differentials, we're kind of one and done. So if we think about Clostridia, we think about other things that we're testing, you're either positive or you're negative on that first sample, and we don't go back and look again. And, you could argue benefits of that, reasons for that, but those tend to be they are one and done. So in that scenario, it's then what happens next. It's that follow-up testing where we know for Salmonella that it's an intermittent shedding and that we're going to have to look at that horse multiple times. And the same argument could be made for herpes, actually, I think, and Dr. P could jump in there, but, the potential for intermittent shedding there as well. And so for Salmonella, I'll stick in my lane, which is, GI disease. But for Salmonella, we're going to want to look at that horse at multiple time points over different periods. And at those subsequent time points, we don't really care anymore about the things that we've ruled out. We're now looking to rule in, is Salmonella being intermittently shed? So that's scenario one. Scenario two would be when we start expanding our biosecurity down into the next risk group of patients, which are our medical colics, and screening our inpatients. The only pathogen that we're really focused on from a gastroenterology point of view is Salmonella in those patients. That's the one that we're concerned about. So that's where a singleplex test comes into value. So there's a scenario, the traditional scenario, where the multiplex still has strong value at initiation, and it's then what happens next. And then you go down into those next population groups, and it's about what's the pathogen we're worried about? It's salmonella. Let's test for that pathogen.
I absolutely agree with Dr. Sykes. It depends. All right. We also have to consider that a single test is, and correct me, Eric, if I'm wrong with that, and Andre, the way the technology is set, when you run a strep equi test on a microfluid card, it's not one test. It's 6 tests. Am I right, Eric? So it's because you have 6 wells. There's 6 wells. So you run 6 analyses. So it's not just one, it's 6 analyses, and 2 need to be positive. 2 wells need to be positive to call it a positive. If you have multiple pathogen on the platform, you're not going to run 6. You may run 1 or 2. So running the single one has greater diagnostic accuracy, in my opinion. And then you have to also put it in the clinical context. I mean, if I see a horse with, a young horse and maybe a draining lymph node, or I scope the upper airways and gall pouch empyema, probably no need to test for any viruses. I'm going to go for my strep equi right away. That makes more sense. And then the other part is, cost, and I cannot speak to that. But if you would tell me that the cost of a panel is equal the cost of a single, then I would say, and it's similar in the production, then probably the single test has little value down the road. But I'm assuming that there will be cost difference, and then trying to be, cost conscientious when it comes to, following up cases once I know what we're dealing with, or if I want to, test cases with a specific clinical entity. I think it's going to become a little bit, with the respiratory pathogen, I think we can get away based on population, time of the year, and clinical presentation. It's going to be a little bit more difficult if you're looking at a, single enteric panel versus a single pathogen for a single enteric pathogen versus a panel that encompasses a variety of enteric pathogens, especially like in falls. I mean, I'll be heartbroken to say, well, I'm just going to test for, coronavirus or rotavirus, or maybe I should have taken C. diff, or maybe I should have taken salmonella. That's going to be, I think, a little bit more tricky. But again, I think it's amazing to have the capability to do single tests for follow-up or to do panel. Can Eric, do you think like cost-wise going to be a big difference?
I mean, in terms
I don't want to,
yeah,
log you
no,
to something that
think,
you'll regret saying, but
yeah, I think for us, we want to, it's all about access, right? So, we've looked at what the reference lab cost is, and we're trying to be, somewhere there. So there's not any additional burden, with our test, right? So, we should know, in a little bit, and have that cost a bit more defined, but yeah, and I think, but the cost is, I think what you're bringing up is actually probably some of the pushback that we get, right? It's kind of, hey, we're already charging the client X amount, you know, 300 or whatever it is for a respiratory panel, and then now we're going to have to add on another 100 for the single plex. But I think what we're trying to do with the practice is kind of understand the different situations you would run each of the single plex tests in, and then understand, hey, what happens if you get a negative? Will you still send out a panel? Which I'm all in support of that. I don't think that point of care is a replacement of the reference lab. I think it's meant to act in conjunction with each other, and it's about getting a result in 1 hour quickly, and then you have, let's say at Davis, right, you'll get that result in 5 hours, and then maybe you're somewhere else and you get that in 2 or 3 days. But I think it's really about looking at your algorithm and saying, okay, in the screening test, maybe I don't need to have that. It's a cost plus, and then you're getting your value from running the panel. Or in much more obvious clinical situation, that's where it's maybe warranted to have a little bit of a higher markup or a stat testing on the weekend or at night, right? So I think that's something we're trying to be a little bit better with, in terms of supporting our practices, and like, how do we financially look at the whole, let's say the whole process or the diagnostic algorithm, and how that gets communicated to the client at the end of the day, so that there's no surprises.
And I think it's building in that. Sorry, Dr. P, go ahead.
I've never found that any test is too expensive. I always found that the markup is sometimes too high, which is two different things. And I'm the first one to start screaming when our hospital increases the charges 5% for CBC or biochemical panel. But then I realized there's no markup. We're probably the only academic institution, the only facilities that, the resale or the charges for a test, it is what it is. It is what it's cost. So there's no markup. And I know that in private practice, the markup can vary from nothing. If you feel like that's the way I look at it. If I gain information from a biosecurity standpoint and gladly do not put that horse that not tested positive for strep equine in the middle of my, hospital wards and I put it in isolation, that's good information for me. I'm happy to sell it at cost, but I don't have to make a living by selling tests. So I understand why tests are marked up. There's work to it. There is, interpretation to it, but the markup needs to be appropriate, I think, and, according to the case. And I know there's a lot of variations there.
Yeah, and I think there's two things that come out of that. So part of that is that that role of the tests aren't being done in isolation. It's like you use the example, the lymphadenopathy with a snotty nose. Why are we testing for viruses? It's really not very high on our list. Looks like a duck, walks like a duck, and quacks like a duck. What we're primarily interested in is whether it's strep equi or strep equi zoo, in most of those cases, or some other bacterial pathogen. And so I think that's where, the singleplex starts to really play a role as well, is in, once you've refined that clinical question and asking actually what bacterial disease do I think this is, or what respiratory disease do I think this is, viral disease, then they become, much more targeted and much more diagnostically useful. And the other is the value of the negative. So, Dr. P, you mentioned, you're happy to, if it tests positive and it ends up in isolation, that's great value for your hospital. If it tests negative and it stays out of isolation, that's great value for the client. And not knowing that on a lot of those horses early on, when they come into the clinic, I think that they're often the respiratory ones are the hardest ones to decide whether they end up in isolation or not. And knowing what they're not can be just as useful, I think, sometimes, and really change your risk perception, change whether that horse ends up in isolation, and then change the overall cost of the client. So I think it really loops back to that same central tenet of the test not being used in isolation, the test being used as part of that decision-making criteria for outcome A, B, or C.
Yeah, I think that's but I think it is a complex concept, and maybe that's something we can work on too, is how do we kind of prepare our vets to have that conversation with the client too, and just decide on those things. And I think we're prepared to do that, but it is something where, let's say, I don't own a horse yet, but I would want to know how many.
Well, if you get one, you have to get two, Eric. You have to have two horses. You're not allowed to have one.
I have two cats. I have two cats.
Well, and I think it's relevant, when we think about from an internal medicine perspective and pharmacology, always sort of try and teach students and vets and whoever we talk to, we're not drug jockeys. We're not just throwing drugs out there. We're clinicians who dispense medications. And I would look at testing the same. We're not just test jockeys. We're not just collecting samples to just mass effect information. We're clinicians who are applying tests logically and sequentially and, factoring in the patient in the decision making. And I think some of our screening tests, they have value at times for screening, but other times they sort of can work a little bit against us, where particularly when they're not 100% accurate tests, sensitivity or specificity, we can actually end up down the wrong garden path if we're not going through that clinical reasoning diagnostic process on the way.
So I've been told if if there is a question, you can add to the chat, or apparently you can be unmuted. So not exactly, I'm not the master, the Zoom master here, but that is what I've been told. So feel free to continue to add questions, everybody. I think I think this other question here is actually a pretty valid one. You know, primarily we're looking at, a lot of our users are more on the referral hospital side, or, you know, maybe have a little bit of a larger facility. You know, do you guys have any maybe advice for groups that are more ambulatory? This also could be from our application scientists too, you know, in terms of testing for respiratory disease and enteric pathogens. But yeah, ambulatory.
I'm going to jump in. My thought is, as long as you have the sample collection kit and the fridge, there should be no barrier to collecting a sample and processing it as soon as you come back from the field. And that still beats having to wait an extra few days, sending out a panel or sending out a test after collecting the sample, waiting for the FedEx guy, waiting for shipping, waiting for the result. It's still quite a good solution in my view.
Yeah, and I think this is where we start drifting from the principle of biosecurity to the application in patient management and clinical decision making. So, you know, being able to identify our pathogen to guide our therapy and the value of that information more rapidly, where we potentially can reduce our use of other medications such as antimicrobials. I think that's where the value starts coming, and building that value proposition in to the discussion where, well, if we can get this result today, we don't need to start antimicrobials now. If we can get a result today and it'll guide us one way or the other, we know it's this, we can go down this direction. We know it's not this, we can go down this direction. And I think that's where the value starts coming in from an ambulatory perspective.
Perfect. Yeah, I agree. I think what also I've seen is maybe, you know, is on the veterinary side, you know, how do we leverage your expertise for your clients that are, you know, maybe have their own facilities? I think, you know, we want to leverage our team to help you improve the biosecurity of your clients. So that's also something where, you know, you may not have the solution right now, but you know, I think we have a team around us and with our users as well, where we can help you do that. You know, our goal is just to keep improving biosecurity, and there's definitely a lot of facilities out there that I've encountered where it's, hey, I think there's additional layers we can add with this type of technology. So again, testing doesn't solve everything. It's one piece of the puzzle, but I think, you know, it's really about in spirit of the application science and, you know, our advisors here, and I think how do we craft something that you can take to your clients as well?
I think the application for ambulatory veterinarians are there in a biosecurity standpoint. I mean, if you look at all the outbreaks, somebody defines the outbreak and recognizes the outbreak. That's generally not somebody in academia or a large veterinary hospital, am I right? I mean, that happens sometimes. You get the referred case, but generally the outbreak happens in the field. So it's recognized by an ambulatory practitioner, multiple horses, whatever the syndromes are. And then you go and you test. And as always, and somebody needs to do the study on it and explain why all outbreak happened on Friday afternoon. But that's a reality. They happen at times where your official laboratory is not open. So what you do, I mean, independent of your result, you're still going to do and proceed to use good biosecurity protocol. But to me, having access, and I realized that maybe, you know, 2024, as an ambulatory practitioner, having Fluxigy, PCR, instrumentation in the back of your truck, probably not gonna be a common finding. Am I right? Just because of the application. But knowing who has one right now, knowing that, the hospital that is maybe 15 minutes or half an hour, even an hour away from where you practice has Fluxigy instrumentation and is willing to run your sample, I would go for that. Now, once, this technology is multimodal where you can run serum amyloid A, acute phase protein, cytometry, individual, metabolite, renal panel, that's going to be a different ball game. And I foresee the future for an ambulatory practitioner is to be able to on-site run whatever you decide you want to run for the management of your patient. But until then, I think that, having a PCR, just PCR instrumentation out there in the field, it's going to be a hard sell. It's the same with, X-ray machine. But if you can share it, that's the other concept. I know that veterinarians don't like to work with other veterinarians. Just kidding on that one. Maybe I'm just talking about myself. But imagine if you work in an area where there's various ambulatory practitioners, and you say, yeah, I see some cases, you see some cases, and Johnny sees some cases too. Hey, let's get one instrument, let's place it at somebody's home as long as we have 24/7 access. Why not? I think there's different models that we can think about it. And the way I look at it, this technology is only gonna diversify over time, where it's not going to just be PCR. I always tell Eric, you have started with the most difficult application and the most difficult pathogen. But if you can overcome that, the doors will be open. Running a kidney panel is going to be peanuts. Running an inflammatory panel cytometry, that's going to be peanuts. That's not a challenge. That's reality. They're going to be establishing that. It's just they decided to start with the hardest one, and I'm glad they did, because they have overcome a lot of burden and a lot of challenges, and they've built resilience and definitely a better product.
And that segues into
we have one question too here. I don't know
that segues into Dr. St. Laurent's question, which is the ability to separate the machine from the samples. So the question there is, ambulatory vehicles, you've got lots of vehicles, but not necessarily lots of machines. You can certainly centralize the machine and bring the samples to the machine. And maybe Andre or Dr. P might want to comment on, the relative stability of the samples, because we always talk about it as, come back and run them that day. You can run them the next day. You can, there's, I'll leave you guys to fill in the specifics, but that creates a lot of flexibility to do it that way. Andre, do you want to comment on the stability of the samples, how long you can wait before you run them?
I can comment on that. Ideally, we want to run them immediately. However, if they're placed in VTM, they can go up to 72 hours at refrigeration before the sensitivity starts to take a hit. Now, for putting them in red top tubes without any buffer, I personally do not have that data to comment on. However, I just, as somebody who has run a few tests, I will say that if it's within the day, if it's kept cool, it should probably work. However, it's not something that we've run tests on.
It's I think that the rayon tip swab is important there. That is a synthetic polyester type swab, and I think in that it seems like that's what's been continuously submitted to UC Davis all the time, and so yeah, I think you'll be fine. Again, like with the refrigeration, as long as it's not in the heat. I think we should send Dr. St. Laurent some collection kits though. As a reward for sitting through the office hours. I'm not sure who our account manager, but I'm just gonna put that out there. I'm gonna get in trouble, but also I think we gotta reward, give rewards out for sure.
It's a great question, because I think it really, and it goes back to this. It's not that one size fits all. It's that creating adaptability to make sure that these different scenarios get encompassed, because everyone's going to have a different set of drivers and a different set of resources available to them, and making sure that comes out in the conversation. So I think it's a great question.
we'll send a little bit of a more comprehensive response as well to that question, that in terms of the stability. But yeah, I think in red top tube and then refrigerate, and then you can add to our BTM before you test is fine, as long as the swab is polyester. That's
No cotton tipped swabs.
no cotton, no wood applicators. If we can send Dr. Saint Laurent a comprehensive email, that would be awesome. Last few minutes here. Any comments? Any last comments here? I think.
So I'm gonna add something very controversial. No, I'm just kidding. You have all these requirements, am I right? I gotta be Ryan tip, and you gotta have the valve transfer medium, and be sure the right temperature, and don't shake the package too much, and be sure to use FedEx instead of UPS. I'm just kidding. We have all this restriction at the end. You're like, yeah, whatever, forget about it. That's way too complicated. So if the only thing you have is a Q-tips and a red top tube and an ice pack, please do it. That's the short version. It will still work. This is PCR, and we're dealing with DNA pathogen so far. So I'm not too worried. It's still gonna work. Now it's gonna be different once we start looking at RNA pathogen. RNA is not very stable, but the key element is really refrigeration. If you can refrigerate a sample, doesn't need to be fancy, just an ice pack. In doubt, just freeze it and then ship it. And if it's synthetic, great. If the only thing you have is cotton swab on the wooden shaft, well, the worst thing that can happen, you get an error because you get inhibition. But at least you get an A+ for trying. The other thing is everybody has a culturette, am I right? The little culturette with the charcoal at the end. So the swabs for the culturette are made out of synthetic material, Ryan tape. So everybody should have one. If you have uterine swab, a little bit expensive to collect nasal swab, but for the occasional collection, that would still work. So there are some alternatives to that. And in doubt, you can even take a 4x4, like just like missing my words today, little 4x4.
Course.
Thank you. I need help, and it's Thursday. Wait until Friday. I'm glad this meeting is. So get a gauze, shove it up the nose. Don't lose it, but shove it up the nose or wipe the nares. That works as well if you're looking at respiratory pathogen. But again, I'm going to deny that I was on this webinar anyway. So for what it is worth, there's a lot of options there, alternatives. So you got to MacGyver it sometimes.
Get some snot.
Just get some snot. Yes.
Just get some snot.
Yeah, I think Marielle has a great point there, where you get an error, then you have to pay for another test kit, so you just lost money. So
just bill it to Dr.
Replace
P.
your test kit, Dr. P.
Yeah.
You are okay.
Send in the bill with an appropriate marker.
Yeah. Yeah, I think no, I mean it is a good point in terms of the errors and the invalids, and I think if you pester your account manager enough, then we can make something happen probably. But I think let's collect the sample first to run the test, and then we can get there. We want to make sure everyone's happy using the product as well. So okay. I think this was a successful office hours. Sorry, everyone was muted in terms if you wanted to jump in, but excited. Please give us some feedback in terms of topics for next time. Excited to keep this going. We'll keep it going, and I think this is just something that we can build on and grow. So thanks again everyone for participating today, and have a great evening, and we'll talk soon.
Thank you.
Cheers, everybody.
Thank you.
See ya.